think we may start now. Okay. So good night, good morning, and good afternoon. So welcome to the our IES microplastic webinar series. Lecture series. Today's speaker is Professor Denise Mitrano. She's assistant professor at the ETH Zurich in the Environmental Systems Science Department. As an environmental analytical chemist, her research focuses on the distribution and impacts of anthropogenic materials in technical and environmental systems. She is particularly interested in developing analytical tools to systematically understand the mechanisms and the processes driving the fate, transport, and biological interactions of particles such as engineered nanomaterials and nano and microplastics. Her work has recently been recognized by Swiss National Science Foundation, Heim Vögtlin Prize for Outstanding Young Woman Researcher of the Year at 2022, and the 2022 James Morgan Early Career Award for outstanding contribution to environmental science from ES&T and the ACS Division of Environmental Chemistry, and the 2022 Emerging Investigator in Atomic Spectroscopy Award. Thank you, Denise. Maybe you can start now.
Great. Thanks very much for the kind introduction, and thanks everybody for being here today to learn a little bit more about microplastics analytics. So as introduced, my name is Denise Mitrano, and I'm Assistant Professor of Environmental Chemistry of Anthropogenic Materials here at ETH Zurich in Switzerland. So when I was asked to provide this lecture today, it was suggested that I go over the typical ways that most people are analyzing microplastics today, because I know many of those in the audience have a mixed experience with already working in this topic so far. So before I get into actually how we measure microplastics, I'd also just like to mention a little bit about analytical chemistry in general. So a lot of folks who are not very familiar with analytical chemistry might think that you can just put in a sample and into a black box, and then you get your answer. But I think that things are a little bit more complicated than that in many cases. And so the reason why I like really developing these different analytical tools to be able to measure different types of particles like nanoplastics, microplastics, and engineered nanomaterials is because it involves a lot of problem solving to try to figure out the best way to develop this method, and being able to have a precise method to be able to measure the characteristics of the materials that we want with a good precision. It's also nice that we can have creatively using different types of conventional approaches in new ways, which is something that is really hallmarked by a lot of the work that we do in our lab, but also developing new tools and approaches to help us better able to solve the questions in our fields. So I think what's particularly nice is that when these different techniques and approaches are developed, it can hopefully be used by more people in the field, and it can really extend our contribution to the field if we develop these really strong analytical methods. So when we think about what we're trying to do when we're actually measuring particles, we first have to think about what it is that we want to measure. So what are the different characteristics of the microplastics and nanoplastics that are important for us to understand in the environment or in biological systems? We have to think about when and where we're going to measure these different particles, because they can change concentration in terms of time or place. And so if we don't sample appropriately, then we're not going to get a correct answer about how much microplastics or nanoplastics are in a given location. And finally, we want to know how we can really measure these particles that are really heterogeneous. So if you've attended some of the other lectures in this series before, you'll know that plastics and microplastics are a really diverse suite of contaminants that are lots of different types of polymers, different sizes, and different characteristics that we want to measure. So how can the different analytical tools that we're going to use distinguish between this heterogeneous group of contaminants? So I would also like to start off with a few spoiler alerts for people that might not be so familiar with the fields. There's no standardized methods currently in microplastics and nanoplastics analysis. So when you start to read papers in this field, you'll see a lot of people using a lot of different approaches. Sometimes they're similar, sometimes not. And then that's also a big question for us in the field about how well aligned these different analytical tools are to be able to give us a correct result. The second spoiler alert is that nanoplastics are really challenging to measure. So the smaller the particles are, the less mass that's there, and it makes it even more challenging to be able to measure nanoplastics compared to microplastics, which themselves are already quite challenging. As a final spoiler alert, it's also not just challenging to measure microplastics in a field setting when we start doing environmental monitoring, but it can be equally challenging to measure microplastics even when you're working in a lab-based experiment. So that's why developing these analytical tools are quite important, no matter where it is that you're working for your different types of experiments. Now, these might all sound negative, but of course, there's some good news on the horizon as well, is that there are a lot of working groups dedicated to developing new analytical tools currently to try to harmonize these methods and also get more reference materials so that we can better work in the lab with more confidence about the results that we're going to get. So now when we think about which types of materials that we actually want to measure, as I already alluded to before, plastics are not all the same, even though we have this big umbrella term of plastics. We have different types of polymers with different plastic added chemicals, and depending on how these plastics get into the environment, you might have different amounts of polymers in one location versus another, and also different size fractions as this macroplastic waste through UV and mechanical degradation comes into smaller and smaller size fractions of microplastic that's less than 5 millimeters in size, or nanoplastic, which is less than 1 micron in size. So now we have all these different types of polymers. So what are the different types of properties that are often investigated and reported by environmental scientists? So what are the types of things that we actually want to know about this really heterogeneous group of contaminants? First, we would like to know the particle size, and then the particle composition, so which polymer is inside, the morphology, so is it a fragment, sphere, film, fiber? These can all have implications on how the particle is moving in the environment or interacting with biological systems. And then, of course, we also would like to know the concentration in our sample. There's two different ways that you can think of concentration when you're looking at particle analysis. You can either look at particle number, so how many particles are there, or as a mass-based concentration, so the total weight of polymers in a given sample. Both metrics can be of interest. I think it just depends on what is your analytical tool that's going to be either able to tell you particle number or plastic mass, or what is your final application. There's a few other types of properties that are interesting to be investigated, but they're not often reported by a lot of environmental scientists now. So it would be important to know what types of plastic added chemicals are also in these microplastics, because these might also have some biological hazard. We'd like to know something about the surface chemistry. So what is the wettability, the roughness, the surface functionality? Because depending on the surface chemistry, the particles may interact with the environment different and interact with biological organisms differently. We'd also like to look at the different sorbed metals and the aggregation state. So how, if the particle is attached to other different types of natural particles, which are also in the environment. So these are a lot of different metrics, which will definitely change depending on which microplastic source that we actually have. So when we look at all the different types of sources of microplastics in the environment, most of them are secondary microplastics. So those coming from the slow degradation of plastic waste or released of small microplastic particles when we're actually using our products. So this can be soda bottles and bags that are littered into the environment, but microplastics can also be fibers that are shed off our textiles or tire wear particles that are released while we're driving. So again, since these are all different types of materials, that's also going to change how we're going to approach our analytics to be able to measure them, because they will be also in different concentrations in different environments. So how do we go about finding the appropriate analytical technique? We first have the analytical problem. So is the particle that we actually have in our sample, is it plastic? And if yes, what kind of plastic is it? So which type of polymer are we actually looking at? How big or small is it? So what is the size? And how much is actually there? What is the concentration? Currently, there's a lot of challenges to be able to answer these, what might seem like very basic questions, because there is this wide range of polymers with different chemical structures. There's different compositions of plastics, so it can be blends of polymers and different copolymers in addition to additives. This all challenges how we're able to address these analytics. The microplastics, which are in the environment, can be all different types of colors and sizes and shapes. So again, it might not be a one-size-fit-all approach, especially when we also considering the different aging states of the microplastic. So when most plastics get into the environment, when they eventually degrade and fragment into microplastics and nanoplastics, they can, to some extent, change their chemical and physical structure, and that's something that we'd also like to measure. But as mentioned, there's no standard protocols now across the entire analytical chain for microplastics analysis. And so this is why many lab groups approach the way to measure microplastics differently. However, we might have some hints about what an ideal method would be if we could have one. So we want to be able to chemically identify the microplastic. We want it to be inexpensive, fast, with low detection limits in terms of particle number and particle size, that it can measure every shape and in any environment. So of course, this is a really nice, ideal list to be able to have a method that would be suitable to us. But of course, not all analytical techniques are able to give us all the information that we want easily and quickly at the same time. So when we start to think about what metrics that we actually want to measure, is particle size and morphology, the general polymer type, and the concentration. These are the metrics which are most commonly looked at when we do environmental monitoring or when we're looking in a laboratory experiments. So when we think about analytics, there's an entire analytical chain. It's not just measuring the particles themselves. So when we look at that total analytical chain, we first have sampling, so how we're collecting our particles from the environment. Oftentimes, of course, these microplastics are not alone. There'll be a lot of other particles, natural particles, which are in the sample. So there's often some type of sample preparation and purification which is needed so that we can extract the microplastics from the sample for easier quantification. And then here, when we talk about quantification, this is where we'll start to use these different analytical tools to be able to give us this particle size, morphology, and different types of physical and chemical characterizations. So we can work through this analytical chain together to see what different types of approaches are used in the field today. So when we start off with the sampling, there's some things that we need to think about when we're collecting the samples and different considerations that we have to have. First is the sample size or volume. So currently it's very variable about how much of a certain water body or soil sample that people will take. And this will also vary on different environmental compartments. But the sample volume is also not tested for the impacts about the number of particles that could be there. For example, if you take a 1 liter bottle of water from a lake versus 10 liters versus 50 liters, you may get a different answer about the total number of particles that are in there, because it is a really heterogeneous mixture across the entire water body. Something that should also be considered is the sampling frequency. So one time point might not be enough to be able to really get this full heterogeneity of plastics that are in the environment. And additionally, having replicates, so taking a same sample at a given time in multiples so that you can make sure that you have a good analytical result. Now, because the time and expense of actually analyzing the plastics and microplastics is so long, currently there's not often a lot of these kind of basic quality assurance and quality control type measurements when measuring microplastics in the fields. And so this makes the statistics of the particle measures sometimes a little bit challenging to compare across different studies, because overall there's relatively few particles measured of a really heterogeneous mixture. And then this could make some people question how reliable the data is. So when we're actually collecting the samples, there's three major categories, I would say, of samples that we're collecting directly from the environment: water samples, soil samples, and air samples. So for water samples, there are a few different types of typical sample collection methods, including a mantitol, grab sampling, or specially made devices for microplastics collection. When we look at soil samples, oftentimes this is a small mass of soil, either from the surface or through depth. And this could also be if soil cores are taken and sectioned to look at how microplastics are moving throughout the soil profile. But what's especially challenging with soil samples is, of course, there's already a lot of particles that are in soil, and we need to isolate the microplastics from this soil matrix. So we have to have an extraction of the microplastics to be able to actually measure them. In terms of air sampling for microplastics, not so much has been done to date, but there is a big field about looking at aerosols and other particles in the atmosphere. And so this is normally relying on typical types of aerosol sampling. So when we look at water samples, for example, how does the sampling procedure normally look like? So here's some examples of a mantitol, which is in the open ocean, where we have a large net, which is with the mouth of the net is a few meters wide, and the mesh size of the net is about 300 microns. So the researchers will just pull this net from the back of the boat to be able to collect all the different particles that are in the water, and then be able to, again, analyze them. But of course, when the mesh size is only 300 microns, then all microplastics that are smaller than this size are often going to be not captured, and you wouldn't be able to measure them. However, if you had too small of a mesh size, then you're going to have too many particles that are clogging the net, and it won't work either. So this is why some researchers have started to develop specially designed equipment to look at microplastics or collect microplastics from different water samples. For example, if you have different cascading water filtration units, that you can have sequentially smaller size microplastics collected in each area of the filter. It also depends on which water body you're sampling, which type of sample approach is appropriate. So if you want to look at just the surface microlayer, you may have a hand net or something similar. And if you're going to bigger and bigger water bodies, then you might have some of these bulk water cascade filters or this mantitol. When we think of soil samples, generally this is a much smaller sample size of having a few grams of sample of soil that you're actually going to collect. And that's what makes this even more of a heterogeneous sampling, both in terms of spatially across the whole field which you're sampling, as well as the content that you'll eventually measure. So now that you actually have your sample, which may contain some microplastics, we need to get rid of the other particles to be able to isolate the microplastics to be able to measure them. And so this is what we call this sample preparation. So when we have these considerations for extraction, we're trying to separate these other natural particles from the matrix to get our microplastics alone. So there are a few approaches which are typically used in literature. The first is physical separation. So this could be as simple as laying your sample out on a bench and trying to pick out with tweezers your microplastics. But of course, that's very subjective, and you might not get all of your sample that you're looking for. There's flotation, so looking at the variable densities of your inorganic material, like sand is oftentimes more dense than plastics, so we can separate these with a separatory funnel. And for those types of things in the matrix, like organic materials, we can have a chemical degradation of the matrix, either through an oxidation of the organic matter or different types of enzymatic digestions. We may be able to help ourselves a little bit by having some visual distinctions with dyes or fluorescence, but that's more of a post-treatment. So when we start to look at the physical separation, here density separation is typically the most used technique in the field today. So on the left side of the figure, you'll see our soil sample, which has some microplastics, and if it is just in water, the microplastics and the sediment will all sink to the bottom. However, if we put a different type of salt solution, we increase the density of this water, and then the microplastics can float to the top and hopefully be able to be separated easier. So, of course, the different polymers have different densities. So you need to match the correct salt solution, which you want to be able to recover and float your different polymers to the surface. So while some of the salts to use, like sodium chloride, are very low cost and have a low toxicity, they also don't have a very high density, which means you may not be able to recover some of those higher density microplastics. And so that's why many researchers use more heavy salts to be able to recover more dense microplastics as well. However, even if you get rid of all of the sediment, the organic matter and light particles or small particles will also float in this high density matrix. So it's difficult to isolate only the microplastics with this approach because you may still have a lot of organics which are in the sample. So when we think about trying to isolate microplastics even further, we may have to do a second step to degrade the organic matter. So there's two typical approaches which are used in the field today. One's chemical degradation, so using something like hydrogen peroxide to be able to degrade this organic matter, or different types of enzymatic digestions to more softly degrade this organic matter. Because it's quite important that whichever degradation method that you choose to get rid of the organic matter doesn't impact your plastics. Because some of the more aggressive chemical degradation methods may also degrade or further fragment your plastics. And of course, that's not what you would like to do. So we tried to also work through an entire sampling and extraction approach in our lab as well, where we took a systematic approach to have a soil sample that we made a composite in the lab of different types of materials, organic matter, clay, silt, sand, that we mixed together with microplastics, as well as some other lignocellulose, and then tried to remove the different components from the system to get the microplastics alone. So we could then do some density separation, the lignocellulose removal with the organics, and eventually we wanted to make sure that we had a clear filter, so just having the microplastics alone, and ensure that there was no changes chemically to the polymer so that we could really identify the microplastics well. So when we look at the elimination of the inorganic components, we had a density separation, which again, in this case, was enhanced with salt in a separatory funnel. So we could try to get most of the microplastics that are floating on the surface. And then we could try to eliminate the really small size inorganic particles like clay by passing the remaining supernatant through a stainless steel filter. So we would still have hopefully all of our microplastics on this filter, but having less of the inorganic material there. Now, there's still some organic components that need to be removed. And for this, we tried to go with the enzymatic digestions using different types of anaerobic fungi that can degrade this organic matter. So as an example, to show that this worked, we could start with just some organic matter like straw, and then expose this straw to our different enzymatic digestates and Fenton reagents for a reactive oxygen species formation to slowly purify the sample. So from the start, we started with 30 milligrams of straw, and after the entire treatment, we could see that there was no straw left that was in our sample. And so we would also expect this to be the case when we have microplastics inside, so we would be able to identify them easier. So when we look at the entire workflow of how to do this extraction of microplastics and quantification, this is, of course, just one example which we do in our lab. Others take different approaches, but we have our microplastics, which are soil, which we spiked with microplastics. We did a density separation to remove the sand and the silts, a filtration to remove the really fine colloids like clay and silts, and then started to degrade some of these organic materials by using Fenton reactions and enzymatic digestions to remove the organic material, and then finally be left with just our microplastics that are on our filter, which can be then further characterized. So now that we have our sample more pure, then we want to analyze it. There are a few different analytical methods that can tell us a bit more information about these microplastics which are on the filter. So these can be split into two broad categories: physical characterization and chemical characterization. In terms of physical characterization, this is mostly through microscopy. A series of different types of microscopy techniques are available to be able to look at what is the size and shape of your microplastics that are on the filter. But it's often the case with microplastics that you just don't want to see a particle there. You want to know that it's also a microplastics particle. And that's where the chemical characterization is also very important. The chemical characterization is also very important, where then we can get some more detailed chemical information about the particles which are on our filter. We can also look at different types of thermal analysis, like pyrolysis GCMS is also quite popular. So when we see the most popular and common methods explored in the field today, I've highlighted these here in red in terms of both those that are appropriate in physical characterization and chemical characterization. But it's important to also note that some of the different approaches will give you different information. So some of those, like microscopy and spectroscopy, when it's coupled with microscopy, will give you information on particle number. But when we look in thermal analysis and look at pyrolysis GCMS, this is only going to give you plastic mass. So again, it really depends on which type of metric that you're interested in for your study. So if we start with the most basic type of analysis, just visual sorting through microscopy, I'm sure many people from basic biology class have used an optical microscope before. And so the concept is very similar, that you would just have all of your microplastics, which are put under the microscope. And people are just looking for unnatural colors to see if you have a particle which is blue or red or pink or something like this. And then it would be suspected to be a microplastic, or testing it with tweezers to see if it feels like a microplastic. But of course, this doesn't give you any chemical characterization and identification. So it's not a very good and scientific approach to just use optical microscopy. Now, I think it's a little bit less used in the field today, but when microplastics research started a decade ago, this might have been more common, especially because it's fast and it has a really low cost. But there are many disadvantages which are very clear, especially that it doesn't have any chemical identification and that it can be really subjective and misleading with what different researchers are considering to be a microplastic or not. So to be able to give a little bit more certainty about if something is actually a plastic, still using a more basic microscopy approach, others have started to look about using a stain with fluorescence to dye the microplastics so that you would have a little bit more idea that this is actually a microplastic, because the idea is that this fluorescent dye would be able to bind to the microplastic, and then you would be able to measure it easier. And so you can see that also in the figures at the bottom. If you look at just the regular optical microscope, it's a little bit challenging to identify if this is a microplastic or not. But if you have a specific dye which is binding to the microplastic, then you'd be much more certain that this is actually a microplastic instead of another type of particle. So as before, this is also really fast and low cost, or relatively fast and low cost, but still has several disadvantages, especially because a lot of other types of particles, for example, microorganisms, can also autofluoresce. So it could be difficult to actually differentiate between microplastics and other particles. And sometimes other particles can also be stained in the sample. So you may have an overestimation of your microplastics. And the different types of polymers may react differently to different types of dye. So it may not be selective enough for you to be able to use just one dye to be able to look at all different types of microplastics. So this is still maybe not the best approach to really make sure that you have your microplastics in the sample, but still is a step above just using optical microscopy. But of course, it's best if we can actually have a chemical identification. So when we start to look again at this analytical methods, which are typically used, the next one we can look at here is the spectroscopy, especially FTIR, which is coupled with microscopy, is what is typically used most in the fields today. So when we look at FTIR, we have a source that is put into the machine, and then the sample can be excited by this source to be able to give us some further chemical information. So the measured light intensity which hit our sample is the Fourier transformation that has to be then applied into the specific polymer. And the spectra shows the vibration, which is inducing a change in the dipole moment of a specific polymer. And then when you use this Fourier transformation, you're able to get something that is basically like a fingerprint of a specific polymer. And so here you have, for example, polyethylene, but other different types of polymers would have a different fingerprint to be able to say that this is really the material that you're looking at. So when you couple this type of technique with a microscope, this is something that you may typically be able to get. So this is a typical microplastic sample that has already been purified using some extraction technique. There's a lot of particles there. Some of them might be microplastics and some not. So when this goes under the micro FTIR, you are able to start to see some of the particles which are more likely suspected to be plastics, because the beam is rastering over the entire sample and measuring particle by particle the chemical composition of this particle. And then with a software, we'll automatically identify which of these different particles are suspected to be microplastics, or at least be a polymer. And then the researcher can zoom in to your different polymers of interest, and again, look at this different spectra and be able to decide if this is really a microplastic or not. Now, of course, this is a little bit time consuming to be able to go through every single particle on the filter, but there are a lot of different softwares that are becoming increasingly available to start to expedite this process as well. So when we look at the method overall, it is good that it is non-destructive. It's relatively fast, and it can be relatively reliable because you get this chemical information, especially now that more data sets for FTIR spectra are becoming available for comparison. However, there are still some disadvantages where some types of particles are more difficult to analyze than others. It can be relatively expensive, and you need to have quite some training to be able to interpret these different signals as well. And depending on the environmental matrix that you have, it can make the spectrum more difficult to interpret, especially if the particles are covered or around other natural particles that are not polymers. Another nice thing about FTIR, though, is to be able to also assess the chemical transformations that happen over aging and weathering of the polymers. So here again, we can look at this spectra that we get, for example, for polyethylene. And in this experiment, we took different polymer films and then exposed them to UV irradiation, which changes the surface chemistry of the polymer. And then when we can zoom in to these areas where we have most of the peaks, we can start to see these changes in the chemical composition easier, and then are able to see the extent of oxidation, for example, on the polymer surface when we have the FTIR information. So the FTIR, micro FTIR is one very common technique. The other quite common technique which is used today is pyrolysis GCMS. So here we're not going to be able to look on a particle by particle basis, but the total mass of polymer which is in the sample. So this is working in a quite different approach, where we can still now take a direct analysis of a complex environmental sample. So it has potentially less sample preparation than needing to isolate the microplastics for FTIR. But there's, of course, some drawbacks as well. So in this approach, you take a quite small amount of sample and put it into a small crucible. You can heat up this entire sample, and then the sample is thermally degraded and measured in the GCMS. And then you can start to see the different fragments of or parts of the polymer, which are measured in the output. So this way you can also start to see the characteristic, again, simplification, fingerprint of the different types of polymers, and then be able to start to see which types of polymers are actually in your sample. So when we think about the advantages here, that we can also get information about different plastic added chemicals which are in the sample as well, not just the main polymer, which is typically the case with micro FTIR. And it could be considered a less tedious sample preparation. But a disadvantage is that we have a lot of information loss in terms of particle size, shape, and particle number. We only know the total mass of plastics which are there. So again, it really depends on which type of information that you're looking for in your sample, if you would choose to do pyrolysis GCMS versus something like micro FTIR, which would be able to give you these different pieces of information. The sensitivity is also quite good, but you still have a relatively high mass of sample, which is needed as a high mass of polymer, which has to be in your sample. And there can also be some interferences from different parts of the environmental matrix as well. So these are the most common types of microplastics analysis now, but there's now, I think, is a really nice time to look at new and different approaches in the fields, which might be able to help us answer different types of questions. So in our lab specifically, we don't use too many of these, let's say, traditional or typical microplastics analytics, but trying to explore new approaches, which would be able to look at microplastics in environmental samples. So when we look at the particle analysis, we look at things like flow cytometry or ICP-TOF-MS to see if these tools will be able to give us additional information in a more high throughput way than is currently possible with the FTIR, Raman, or pyrolysis GCMS. We are developing tools to look at the absorption of biomacromolecules on polymers, to look at eco-corona formation or if eDNA is absorbed onto the polymer. And for example, we do this with quartz crystal microbalance or fluorescence microscopy. We try to also look at how microplastics are changing the soil structure when they're in the soil by looking at different optode imaging and CT, and following particles in the environment by using different cameras and lasers, and at least in a laboratory context. So there's a few important notes overall when we think about microplastics analysis in general, that there's a lot of possibility for errors and especially contamination at every step in the analytical chain. So of course, in every lab, we're also using a lot of plastics. And so that's why we have to really work in a very clean environment. Everything also takes a really long time, so you can't underestimate how long it actually takes to measure a single sample. And this is where researchers also need to balance the number of samples which they want to measure and to see how if they can really test their objectives or hypothesis with what is actually feasible because of the length of the measurements. And finally, there's not many or really any commercial labs to be able to measure microplastics in samples. And because it takes very long to be able to measure, it's unlikely that, you know, a colleague from another lab would be able to quickly analyze your sample. And so I think that understanding how to be able to do the measurements yourself is really quite important. So now I've given several different types of examples of analysis. So when you're thinking to yourself, okay, great, there's a couple of different options that are available. How am I actually going to choose my analytical methods? So where do I start and which technique do I actually use? I guess most obviously you have to just see what is in your lab in terms of instrumentation, or within your university for instrumentation, or through collaborations. But also what metrics are really important to you. Do you want to know the size and the number, or do you just need to know the plastic mass? And what is the level of precision that's actually needed for your specific experiment? So before you begin with these type of experiments, it's good to understand the pros and cons of the actual approaches that you're going to be able to use, and understand what each technique is actually going to be able to tell you to help you avoid these blind spots when you're designing your experiments. And then after the analysis, when you're looking at the data, see if all the results from your various analytical techniques are actually complementary, and just make sure that you're within the detection limits and resolution of your chosen technique so that you're not over-interpreting the data. So I think that you may have recognized that as I was presenting a lot of the analytical tools, I said that there are some advantages, but many of them still had a long list of disadvantages. So there are a lot of challenges of detecting nano and microplastics in environmental and biological samples. And that's partially because of the heterogeneity of the different types of microplastics which are in the environment now, as well as the current analytical workflow. So it's very time consuming. There's no standardizations of materials or methods, and there's often high detection limits, both in terms of particle size and in the number concentration. So because of this time-consuming analytical workflow, this also gives a lot of implications for how we can think about how microplastics are behaving in the environment, because we often can only have limited number of samples because it takes so long to measure them. And this makes mechanistic studies very time-consuming. And consequently, we have a reduced understanding of basic processes of microplastics movements and biological uptake, even when we're looking under laboratory conditions, because we still have to go through the same current analytical workflow. So in that way, it's difficult to make a risk assessment in terms of both exposure and hazards of plastics and microplastics in the environment now, because there are a lot of analytical bottlenecks which we have to overcome. So there may be, though, like an alternative approach to be able to measure microplastics, especially when we're working in only a lab-based context. So when we're working in the environment, we have to take this environmental sample and measure what it's there. There's basically no way around that. But when we're working in a laboratory system, we can develop these alternative approaches to use model materials, which can make the analysis easier. So there's a few types of these alternative methods where we try to dope the microplastics with a certain dopant to be able to measure things easier. So I already discussed a little bit about fluorescent dyes. So when working in the lab, you can use and purchase already commercially available fluorescent microplastics to be able to use. But there has been some discussion that sometimes these fluorescents leach out of your microplastics, and so may or may not be appropriate depending on your experimental conditions. In our lab, we have started working a lot with metal doping of the microplastics to be able to put a trace metal in the microplastics when we synthesize them, so that we can measure this metal as a proxy for the plastic in our different laboratory experiments. And some other colleagues have started to use carbon isotope labeling, so using C13 or C14 labeled polymers when they're actually making new microplastics, so they can follow this, this radioactive carbon through their environmental system. So regardless of which of these different dopants that you might put into your microplastics, there's a few must-have. It needs to be a unique detection of the plastic compared to other particles which are in the system. It needs to be a conservative tracer, so that means the tracer that you choose has to actually stay with the particle and not leach out or be destroyed. And it shouldn't change the model microplastics too much, because this might also influence your results. So we still want to have the surface chemistry similar to what is a real microplastic, the density similar to what is a real microplastic, etc. So as I mentioned, the approach that we use most in the lab is this metal doping. So I'd just like to go through that with the last 5 or 6 minutes or so of this presentation. So why in our lab do we heavily focus on this metal doping of microplastics? Because there's a lot of potential for this method. So the metal can be used as a proxy to detect plastics, because there's a lot of standardized analytical tools already for trace metals analysis. When we're trying to synthesize these materials ourselves in the lab, we use an environmentally rare metal for the doping, so there's a low background signal. And this makes any type of material, any type of analytical tool, which used to be suitable only for metals, now also suitable for plastics. And with working with colleagues that are more in chemical engineering or material science, we can really produce a lot of different types of particles with different metal dopants, with different polymers, sizes, and morphologies, so that we have a large suite of materials to be able to work with. So for example, we have had this alternative approach of synthesizing our own metal-doped plastics of either nanoplastics, fibers, or fragments. We can then put these materials into a given test system and then analyze the metal as a proxy for the plastic. So using this approach, we have several improvements. It's much faster, it's much more precise, and we can measure smaller particle sizes, including nanoplastics, which otherwise is largely not very possible. And most importantly, we can test many experimental parameters under controlled conditions in a laboratory setting, which makes process studies really possible to be able to better understand the risk assessment of these as we're assessing it in a laboratory context. So the utility of these metal-doped plastics for nano and microplastic research can be put into a few different categories of types of studies. So one is looking at the fate and transport across lots of scales in the lab, so laboratory, pilot scale, and mesocosm studies. We can look at biological uptakes of particles, how particles pass biological barriers and trophic transfer, and using an assessment of sampling and extraction protocols, because we're able to measure the plastics much faster. So the analytical approaches which we often use are all, of course, metals-based approaches, because this is what we're actually measuring, is the metal content as a proxy for the plastic. So the most typical approach that we have is this bulk metals analysis by ICP-MS, where we take a sample, completely digest it through microwave acid digestion, and then look at the concentration of metal as a proxy for the plastic in our sample. We also have more advanced techniques like single particle and single cell ICP-MS, laser ablation ICP-MS, TEM, EDX, micro XRF, and XFI, or basically any other method which is suitable for detecting trace metals can now also be applied to these types of model plastics which we're producing. So how are we actually using this in our own work? We are looking at how microplastics are released from pilot scale wastewater treatment plants in the laboratory, looking at how they're transported down flumes in the lab, looking at aggregation and settling rates of plastics, looking at uptake of plastics to a lot of different aquatic dwelling organisms, looking at drinking water treatment plant purification by using a pilot scale drinking water treatment plant, looking at runoff of microplastics from model agricultural fields, transport through porous media, uptake into plants, and looking at uptake into soil dwelling organisms. So there's a lot of still in the laboratory ways that you can assess this fate, transport, and biological interactions of microplastics and nanoplastics by using these metal-doped particles. So to give you a few examples of how this looks like in plastic, in practice, we can look at the total metal content by complete digestion and ICP-MS analysis of our metal-doped plastics. This is how we conduct most of the studies. So, for example, here we have a column which is mimicking a drinking water stage in a drinking water treatment plant. At the bottom, we can flow through our metal-doped microplastics and then over time collect different aliquots of liquid coming through the top of the column and then measure the metal as a proxy for the plastic to be able to understand how fast the nanoplastics or microplastics move through this porous media. And so in this way, we're able to start to get a breakthrough curve of how nanoplastics or microplastics can move through this porous media. We're here on the x-axis, we have the pore volume of our column, and on the y-axis, we can see the concentration of nanoplastics, which was released from the column compared to the total input concentration that we have. But what I think is most important in terms of analysis here is the number of data points that we were able to get. So in a very short amount of time, we could do triplicate experiments with many data points, which just wouldn't be possible if we had to use some of these more traditional microplastics analytical techniques. We can also have the same idea about taking a sample and digesting it to see where the plastics are. If we start to look, for example, in how plants can uptake nanoplastic. So here we grew wheat plants hydroponically in a solution which contained nanoplastics. And then after the exposure, we could digest either the roots or the shoots to start to see how much metal content there was in each of these different parts of the plants. And then we would be able to see what is the uptake and transfer factor from roots to shoots in this plant. We can also use other microscopy coupled with EDX, which is a metals detection in microscopy, to search for these metal-doped nanoplastics in tissues. So, for example, we were involved in a study where we had these metal-doped plastics, which were given as an exposure to oysters over different amounts of time. And then when these oysters were sacrificed at different times and sectioned, we could then, again, find these nanoplastics inside the tissues, and we could confirm that it was really our nanoplastic and not some other particle because it had this metal dopant inside, which we could be able to measure by EDX. So when we're looking at these different alternative approaches for plastics, I said there's a lot of must-haves to already kind of consider if it's going to be useful or not. But I think that they are quite useful in a lot of contexts, especially as proxies in lab-based studies, which can really solve a lot of the analytical challenges with nano and microplastic detection. But the choice of the proxy that you use, if it's a fluorescent dye, metal doping, or carbon isotope labeling, can really depend on the aim and scope of your study. Although it's not really applicable to environmental plastics because not all environmental plastics have the same amount of metals. If you're going to be anyways using model plastics in a laboratory system, then this might be a really nice approach to be able to take. So with that, I would like to, of course, thank my team for helping out with all of this really nice research that we've been able to do over the last years, and for you for your attention. I'd be happy to take any of your questions.
Okay, thank you. Thank you, Denise, for the very thorough for the introduction for the analytical part of this microplastic. It's very good. So now for question. I think people already asked a question. Can you? Or they said there's no standardized protocols in the field. How long do you think it will take before we start to see standardized or widely accepted protocols? Yes.
Yeah, I've read the question. So thanks, Sarah, for the question, although it's a really difficult one to answer. So I do know that there are a lot of working groups which are focused on analytical, not just method development, but also on standardization and harmonization. but I think that this will be, I don't know, I don't want to say decades plural, but I would say many years until we actually have harmonized approaches. But I think part of that is also because now we're still in, let's say, a testing phase in the field to see what is most appropriate, what's easiest, what do people have access to. So that's what's also important to consider when developing standardized and harmonized approaches, that you also want to be able to develop something that most people are going to be able to use in a high throughput fashion. So I think that it's still a little time away until we get there.
Right, thank you. So any other questions from the audience?
Can I ask a quick question?
Yeah, please.
First, Denise, great presentation, really great overview. And I have lots of questions, but unfortunately I'm rushing off somewhere now to another meeting. So I'll just stick with one last question. The work you've presented, especially the stuff at the end with doping of metals, seems fantastic and a great tool to use in laboratory experimentation, as you've suggested and shown. Do metal-doped plastics behave different in the biota than non-metal doped plastics?
Yeah, so this is one of the most common questions that we get, right? Of course, when we present this approach. So as I mentioned, the metal that we add is really on the inside of the particle, not on the outside, because it's important that the surface chemistry remains as similar to a real microplastic or nanoplastic as possible. So the organism, or in any environmental situation, there's not going to be metal on the surface of the polymer. We also dope it with a really low percentage of metal. It's about 0.2 to 0.3% by weight. So it's not really changing the density of the material either. So I don't think that it's really significantly changing the physiochemical properties of the materials. But I guess another way to also think of it is that since plastics are so heterogeneous anyways, us adding a little bit of metal is still like within the scope of what a normal plastic might be anyways, because there are metals that are used in dyes, for example, which are already in plastics. Now we're just targeting the addition and doping for a specific purpose.
Thanks, that's great. I feel like it's almost like you wanted me to ask that question.
Yeah. And if you have more questions, feel free to drop me a line and we can find another time to chat too.
Yeah. Well, I'll also say, I mean, you presented a lot of stuff on the MantaNet, and it's a bit like my joke. I say things like sometimes the methods are a bit fishy because a lot of them have come from the marine environment. And thanks, it's good when someone gets it. Because of course, I mean, I appreciate that MantaNet can be used to capture a large volume, but given that it's 330 microns or 100 microns, whatever net size, you potentially lose a large fraction of your microplastics. I don't know, do you have any thoughts on that? I mean, what are your thoughts on MantaNet trawls?
Well, yeah, I guess until somebody gets something better, then it's like really hard to not use it, I suppose. I mean, it is good for, I suppose, a screening. I think some of these like sequential filtrations might be a little bit better especially be able to get smaller particle sizes. But maybe I should have also said at the the very beginning, I'm not working in any monitoring or environmental samples directly. We're mostly working in lab-based systems or pilot scale systems because we can use these alternative approaches. Because I think it's a lot easier. But I appreciate that we also need to find better approaches, especially for sampling when we're actually doing monitoring. But yeah, I think that maybe finding eventually moving towards specifically designed equipment that can help us get these smaller particle sizes would be especially relevant.
Yeah, I agree. I mean, of course, it's again one of the challenges. I mean, capturing a representative sample in the environment, and then once we've got that sample, how do we process it? Mm-hmm. You mentioned fluorescent work, and I have the same feeling. It's potentially useful, except many other things fluoresce. So it's, again, a lot of false positives. But there is some work nowadays with people trying to develop specific tags that will, stick to certain plastics, which I think could be a useful way forward for environmental work.
Yeah, I mean, I agree. I mean, I think that it just needs to be developed more and fit for purpose. I mean, I guess the same as with the sampling techniques, right? When it's like fit for purpose. And I think that what has been the nicest thing for me personally, anyways, is being able to now work more interdisciplinary and work with material scientists and chemical engineers, because I think that they have a lot of knowledge that at least me as an environmental scientist doesn't have, and they wouldn't consider how this could be so useful for us, right? So when I first even brought this idea of metal doped plastics to some of my colleagues to help me develop the recipe, they were surprised that, yeah, this was something that was needed and interesting. So I think that that is where interdisciplinary research can really help, to have people with more of that knowledge base being able to help us develop tools which are specifically suited to what we want to do.
Yeah, that's a great point. And maybe it's because microplastics is something that's very new. It's allowed us to step outside those traditional science boundaries where everybody is maybe helping, or at least can be part of a story in terms of sampling, extraction, identifying, etc. So I think that's a great point. Again, I'm going to say thanks very much. Unfortunately, I have to go to another meeting now. Thank you.
Great.
I'll chat again sometime, I'm sure.
Yeah, that'd be
Bye.
great. Bye-bye.
All right, thank you. So let me ask a question regarding the metal doping method, because the particle size, the microplastic or nanoplastic. The nanoplastic, for doping, is that? So I'm not sure. Okay.
Yeah. I should have just focused the whole talk on the metal doping. I think that's the most interesting part. So we make the different materials in different ways. So for the nanoplastics, it's a bottom-up approach where we do an emulsion polymerization, so that as we're forming the particles, we can incorporate like ionic and metal into the suspension and then into the polymer and into the plastic. For the fragments and fibers, it's a top-down approach where we take pre-production pellets, melt them, and then incorporate metal oxide nanoparticles into the melt and mix it around, and then repelletize and cryomill them to get our different microplastic sizes, or use a pilot scale fiber spinning plant to actually make microplastic fibers. So for the different size fractions, you need to take a different approach, because with cryomilling, it's not very easy to mill down to nano size. And even if some people were able to do this, you're not able to have a high enough yields to be able to work with them on a bigger scale kind of experiment.
All right, thank you. Yeah, I also feel that if in the field, because the sample, the different size and heterogeneity for the sampling, so it's very, very, very difficult. Thank you. So any other questions? Let's see. Okay. If no question, so on next week, also on Wednesday start at 8 p.m. and on AM for the US the East time zone. We'll have the Professor Han Zhongjia from Northwest Agricultural Forest University from China. He will talk about the aging of microplastics, role of free radicals. So see you next week. Okay, bye-bye. Thank you, Denise. Thank you, bye-bye.